Assalamualaikum and salam sejahtera. We are from group KK6. We will present about production of probiotics from yeast. Introduction. Probiotics are living microorganisms that are ingested to alter the gastrointestinal flora for health benefits. The main ingredient in the production of probiotics from yeast is the yeast cell itself, which is Saccharomyces boulardii. The production involved aerobic fermentations, so the oxygen supplies was required. The waste produced from the productions are carbon dioxide and wastewater. One of the usages of the probiotics is helps to solve digestive disorders. Some of the raw materials used in the production are Saccharomyces boulardii, glucose, biotin, and thiamine. Some of the properties of Saccharomyces boulardii are it has optimal pH at 4.5 to 5.5 and optimal growth temperature at 37 degrees Celsius.
Next is the economic issues. The graph shows the demand and supply of probiotics from 2018 to 2026. The calculations also show the deficiency, which is 264.599 kilotons per year, and the plant capacity, 1,561.61 kilograms per hour.
As we can see from the process flow diagram, there are 7 unit processes. The first step is which one of a pure yeast strain of Saccharomyces boulardii will be cultivated in the seed fermenter. Then, the expanded seed is fed into the main fermenter to produce more yeast with a supply of oxygen and growth medium. The third step is where the fermented broth from the main fermenter that contains biomass, which is the yeast and waste such as remaining glucose, ammonium nitrate, and water to the centrifugal. This is to make sure that the biomass is concentrated as the wastes are removed. Then the concentrated biomass are sent to filtration to remove the remaining waste and also to concentrate the biomass so it will only contain the yeast. The yeast will be mixed with lactose, which will act as cryoprotectant to protect the cells of probiotics. The next step is where liquid nitrogen will be added so that the probiotics can be formed in pellets of 4 to 5 mm. Then the frozen pellets will be sent to the freeze dryer. This is for maintain the probiotics at low temperature to limit the damage for freeze drying.
For mass balance, we need to find fermentation of stoichiometric equation for production of probiotics. We determine by using molecular mass of each element and yield of glucose for fermentation, which is 0.5. Then we find alpha 3 in the stoichiometric equation that lead us to find another unknown shown in the box on the slide. The calculations show that equation on the slide is balanced. Then we can continue to find mass balance in the fermenter using formula that show on the slide and using basis of glucose in fermenter, which is 765.189 gram per liter in 2000 liter mel fermenter. By using the formula for biomass balance, we determine unknown R in the equation, which is 8.5026 mol per hour, and then we can find mass balance for other elements, inlet and outlet stream, which is shown in the table. We continue with energy balance in a way to find heat energy in the system. First, we have to find heat capacity and molar flow rate of each component in the unit process. Then we'll calculate biochemical heat of reaction, which is shown in the slide, which we get 2760 kilojoule per mole that needed in the equation of heat to be removed. Enthalpy can be calculated using the formula enthalpy equal to R constant multiplied with integration of heat capacity in change of temperature. Then every molar flow rate of element being added to get total molar flow rate of each stream. Same goes with enthalpy of all elements to get total enthalpy of each stream. And we continue to get heat to be removed in the fermenter by using formula on the slide, which we get 81,357.216 kilojoule per hour. This process is endothermic process because we get positive value for heat to be removed.
The waste that we treat in the production of probiotics is yeast wastewater. The treatments that we use are biological, chemical, and ozone treatment. Other than that, there's also the autoclave sterilization treatment. These 6 environmental and quality acts applies to our waste management.
Next, for thermodynamics calculation, we need to use this formula to find the value of K. Therefore, we need to know the Gibbs energy, the heat of formation, and also the heat capacity to find the value of K, which is equals to 0.6562, and this shows it is less than 1, therefore it will result in a decrease in concentration of the product. Next, we need to find the concentration of all the species by assuming that they are in liquid phase and in either solution. We need to use this formula to calculate the composition of each species. Try and error method are used to find the epsilon, which will be equals to 0.8201, and substitute inside this formula. After that, we can find the composition for each species, and the total of its composition will be equals to 1. For the comparison of molar composition, it is compared between the equilibrium constant K and also the molar composition based on the Maxwell balance. So by this we can see that there are percentage error range from 10% to 13%. This shows that the manual calculation is more accurate due to no assumption is made, whereas the molar composition based on chemical equilibrium is calculated with assumption that could cause difference from the actual value. Next, based on the enthalpy reaction in the fermenter, the reaction is endothermic. So, the value of K and the product can be increased by increasing the temperature. That's all.
Chemical and biochemical properties of Saccharomyces boulardii. Chemical properties. Saccharomyces boulardii has high temperature, acid and basic pH resistance. It also lactose intolerance, has low toxicity, and antibiotics resistance. Biochemical properties. It is eukaryotic, single cells, and has diameter between 2 to 3 micrometer. It also undergo non-sexual reproduction by budding method or sexual reproduction by cell conjugation, as boulardii does not digest galactose and also can produce protein that inhibit pathogenic bacteria and their toxins. Selected cells. This table shows the comparison between Saccharomyces boulardii, Saccharomyces cerevisiae, and the Debiomyces hanseníae. Based on this table, Saccharomyces boulardii is the most suitable yeast cells to use in the production of probiotics because it has optimal growth temperature at 37 degrees Celsius, which is almost the same with the human body temperatures. It also has the highest resistance to heat and low pH, and also has the highest yield of product compared to the other two yeast cells. Nutrient in growth medium. Glucose acts as a carbon source, which is supply the energy for the growth of the yeast cells. Ammonium nitrate acts as a nitrogen source that's essential for the protein synthesis in yeast. Biotintiaminosterols are the vitamin that are required to increase the fermentation rate. Phosphorus was needed for production of phospholipid in the cellular membranes. Calcium, potassium, and magnesium act as a key cofactor that can improve fermentative metabolism of S. boulardii.
I'm going to explain for fluid mechanic part. In the fluid of process, we have to find friction loss, mechanic energy balance, suction head and discharge head, net positive suction head, pump efficiency, shaft work delivered to the pump power, and develop head. In order to find that, we need to list down and calculate all stream properties that show in the table on the slide. Then we continue to find friction loss in the pipe from unit process centrifugal separation to unit process filtration by calculate for each subject like contraction loss at exit of centrifugal separation, and we get 1.321 multiplied to 10 to the power negative 3 joule per kilogram. We added all subject on the table to get total of friction loss and get 7.034 joule per kilogram. Then we continue to obtain shaft work by using mechanic energy balance that show on the slide, which we get negative 152.008 joule per kilogram. After that, suction head being calculated with total friction loss in the pipe before enter the and we get value 0.409 joule per kilogram, and continue calculate head using formula on the slide and get 36.89 joule per kilogram. Meanwhile, for discharge head, we continue the total friction after flow out from the pump and get 6.625 joule per kilogram, and we calculate head, which we get 188.1 joule per kilogram. For net positive suction head available, NPHA, we get 15.68 meter by using formula shown in the slide. And for pump efficiency, we've obtained from table of pump efficiency for every volumetric flow rate in the reference. Then we calculate our volumetric flow rate to get it pump efficiency by comparing to the table, which we get 44.6x%, which the pump efficiency we can get pump shaft work and the pump power, which shown on the table.
In conclusion, probiotics can be produced by aerobic fermentation of Saccharomyces boulardii with ammonium nitrate and glucose as sources, centrifugation, filtration, mixing with the cryoprotectants, and freeze drying. The plant capacity is 1,561.61 kilograms per hour. The main fermenter undergoes an endothermic reaction with the heat of reaction being 2,760 kilojoules per mole. The equilibrium constant is less than 1, so the equilibrium reaction moves to the left, resulting in a decrease in the concentration of products. The extent of reaction is determined, which is 0.8201. However, there are percentage errors when the equilibrium constant composition of each component is compared with the molar composition of mass balance. This is due to the assumptions that all phases are in liquid. S. boulardii strain is selected among other yeasts like S. cerevisiae and D. hansenii because it has the highest yield of product, almost the same temperature with the human host temperature, which is 37 degrees Celsius, more resistant towards a low pH and very high temperatures. The total friction loss from the pipeline flow into the pump is 0.409 joules per kilogram. The shaft work obtained is 152.008 joules per kilogram. The negative sign indicated that this is a work done on the system. Finally, the pump efficiency is 44.68% for 0.02406 meter cube per minute. That is all. Thank you.